Welcome students. Today we will take our polysaccharide, the last part of this carbohydrate. Polysaccharides are basically made up of more than 10 monomeric units. There are more than 10 monomeric units. Polysaccharide, large number of monomeric units, maybe 100, maybe 50, I mean to say more than 10, are getting linked to each other by glycosidic bond and form polysaccharides. These polysaccharides are further of two types. One is, sometimes we call these polysaccharides as glycans. We also call them as glycans. The polysaccharides are of two types. One is the homopolysaccharide, and second is the heteropolysaccharide. Okay. Homopolysaccharides are those polysaccharides in which the monomeric unit is only of single type. We have one type of monomeric unit present in the homopolysaccharide. For example, if I will show you here, suppose this is some monomeric unit, and these monomeric units are getting repeated time and again. They are getting repeated continuously. But how many types of monomeric unit we have here? We have just one type of monomeric unit. Suppose we have started with the glucose, and this glucose is repeating continuously. All the monosaccharide units are made up of glucose, and here the polysaccharide so formed is formed by the aggregation or by the combination of single type of monomeric unit, maybe glucose, maybe something else. Therefore, it is a homopolysaccharide. And heteropolysaccharide may contain two or more than two types of monomeric units. There are two or more types of monomeric units. If you see, so heterosaccharides may be like this. Then we have suppose one type of monomeric unit, and we'll be having some other type of monomeric unit. Maybe in the green, I will show you this type of monomeric unit. Again, we have this type of monomeric unit. Okay. And then again, we will be having the other type of monomeric unit, which will be written in green, and so on. Here we have two types of monomeric unit. This is one type of monomeric unit, and this is the another type of monomeric unit. Therefore, we will call it as a heteropolysaccharide. You may have heard about polysaccharides. Polysaccharides as storage polysaccharides. You may have heard polysaccharides which are the we can say functional polysaccharides. If we see, we have storage polysaccharides. For example, the main important storage polysaccharides we have is the starch, and glycogen. These are the storage polysaccharides. They are abundantly stored. Starch is stored in our body. Starch is stored in plants. As a storage polysaccharide of plants, we have seen starch. Glycogen we are finding as a storage polysaccharide in animal cells, in our liver and in our muscles. So there may be storage polysaccharides. Or we may have the structural polysaccharides. These are storage. And we may have one thing which we call as structural polysaccharides. These polysaccharides are contributing towards the structure. They are making the structure. And if you see the structural polysaccharides which we have is the cellulose. Cellulose forms the structural polysaccharide. Chitin forms the structural polysaccharide. Cellulose is making the structure of plant cell wall, and chitin is making the exoskeleton of arthropods. Again, the structural polysaccharide. So this way we can have structural polysaccharides as well. Now we will discuss these homo and heteropolysaccharides one by one. Some notable examples which are important so far your syllabus is concerned. We have one important polysaccharide which we call as starch. See, starch is the storage polysaccharide of plants. It is stored in the plants, made by the plants by the process of photosynthesis and stored there, right? This starch is made up of only one type of monomeric unit which we call as glucose. And the glucose is in alpha D form. Alpha D, you know everything. This is made up of glucose. And if we see this starch, if we treat the starch with boiling water, if we treat the starch with boiling water, it gets dissociated into two components. One is the amylose. One is the amylose. Treating this with the boiling water dissociates it into amylose, and it dissociates it into amylopectin. Amylose is water soluble to some extent, and it is less water soluble, right? Less water soluble. Second thing is, amylose is showing a structure of alpha 1,4 glycosidic bonds, and amylopectin is also showing a structure of alpha 1,4 glycosidic linkage, glycosidic chain, and a branch point points of alpha 1,6 glycosidic bonds. It means if we have to see the amylose may be like this: glucose, 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 alpha 1,4 glycosidic linkage, linear sequence of monosaccharides, and these linear sequence will be having the similar structure as that of maltose, just a repetition of maltose. But when we talk of this part, it is made up of, yes, glucose, 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 but somewhere we have the branches also. So amylopectin has a branches in addition to the linear structure, it has branch points as well. You have to note it. Then it will give blue color with iodine, and it will give a red color with iodine. If I treat with iodine, giving blue, and this is giving the red color with iodine. And both the two, whether it is amylose or amylopectin, can we digest starch? Of course, we digest starch. We digest it in the mouth, we digest it in the small intestines, right? And there are enzymes in our body who are digesting starch, and this starch is digested by both alpha amylase and beta amylase. The enzymes which digest starch is known as amylase, so we can digest it by both alpha amylase and beta amylase. So far as structure is concerned, amylose and amylopectin, we can write it like this way. Very simple structure we have. In one we have the linear sequence of maltose. We have just to repeat the maltose units, and you know how we can make it. We have this is a glucose, for example, all right, and alpha D glucose. Alpha means OH is here, OH is here, OH is here, OH is here, CH2OH, all right. This is alpha D glucose, and you know this is H, this is H, and this is also H. Again, you make one more alpha D glucose. This time we can make alpha D glucose, one more alpha D glucose. All right, you have just to make the maltose, alpha D, OH on the lower side, OH is here, OH is here, and it is CH2OH. Here it will be H and H. This is alpha D glucose, alpha D glucose. Then what happens? This is a type of condensation reaction where the two monomeric units are condensed to each other. Again, this is position number 1. This is position number 1. This is 2. This is 3. This is 4. This is 5. This is 6. 1, 2, 3, 4, 5, and 6. And here this H and this OH will go, and they will form a bond. You know it, which we call as glycosidic bond. And here also there will be formation of glycosidic bond, and then this process will continue to the other parts also. Number of monosaccharide units will be joining in this way and forming what we call as polysaccharide. All right, this is the structure of starch. Then we have starch, one part of starch, that's amylose. When we have to write the structure of amylopectin, see, amylopectin has the basic frame which is same as that of the amylose. It means this is the amylose. Similar frame will be there for the amylopectin, because amylopectin is also made up of linear sequences of alpha D glucose. We have the sequence of alpha D glucose repeated many times. This will form the basic frame of amylopectin. But amylopectin has one more thing which we call as branch points. See, for example, I will make it like this. We have in the amylopectin, we have one more. We have this glucose unit. One more chain of glucose unit. O and O. This way. Okay. This way. This is O. We have one more chain of glucose units. Glucose, linear sequence of glucose units. Here we have CH2OH. This will be H, it is position number 1. Sorry. I made it wrong. Here we will see it this way. This is another chain of glucose, another linear sequence of, just like maltose we can say. This is O, this way, and this way. It will be continuing here. Here you have OH, and here you have H. At position number 1. What is going to happen? Here we have CH2OH. This CH2OH will present at position number 6, and this OH, which is present at position number 1, will be joining with each other by removing a molecule of water. They will remove a molecule of water, and thus they will be forming a glycosidic linkage between this CH2 and O. The O will remain in the center. This type of bond is known as a glycosidic bond, but alpha-1,6 glycosidic linkage. This is the difference between amylose and amylopectin. In amylopectin, you will get branch points as well, and the branch pointing is alpha-1,6, which you will find in the long chain of amylose, and at after 24 to 30 residues of these monosaccharides. For example, if it is here, branching is here. After the 24 residues have elapsed, after 24 residues, after 24 residues, there will be again one more alpha-1,6 glycoside linkage. I told you basically the repetition of alpha-1,6 glycoside linkage takes place after 24 to 30 monosaccharide residues. All right. Okay, after this we have second polysaccharide, which we call as cellulose. So the starch, or we can discuss before cellulose, we can discuss one more polysaccharide which is called as glycogen. Let us see what is glycogen. पहली बात ये है, this is the storage polysaccharide. It is the storage polysaccharide of animal cells. It is the storage polysaccharide of animal cells. Animal cells are storing this polysaccharide at two places. One is in the muscles and second is in the liver. As a source of energy, we are storing it. And if you see the weight of the liver, so as this glycogen is concerned, the liver has 7% of its wet weight contributed by glycogen. So, liver is the main storehouse of this glycogen. The question is why the liver is storing glycogen, not glucose. The reason for storing glycogen as the principal sugar, storage sugar in the liver, is that it is not increasing the osmolarity as compared to glucose. For example, if the liver will be storing glucose, the osmolarity would have been increased. And what will happen with the increased osmolarity? Once the osmolarity is high, in the cells you have higher osmolarity, you will find more osmolarity in the cells. Large amount of water will come in. It will be dragging the large amount of water from outside, and this will be expanding the cell, and thus it may be also bursting the cell. Therefore, glucose is not a preferred sugar for the storage at our liver, at our hepatic level. It is only glycogen which can be stored because of one thing that's very important, that's the osmolarity, and this osmolarity is very, very less as compared to that of the glucose. So this is one thing. Second thing, if we see the structure is just like this. Glycogen has structure just like amylopectin, but there is a difference. See, when we discussed amylopectin, we have seen it has one alpha 1,4 glycosidic linkage, all right, and it has alpha 1,6 branching. Same is the story with the amylopectin. Then what is the difference between the same is the story with the glycogen. Then what is the difference between glycogen and amylopectin? In case of glycogen, the branching is highly dense. You will find more intense branching in glycogen as compared to amylopectin. In amylopectin, we have seen the branching was at 24. There was a branch point after 24 or 30 monosaccharide residues, and here you will find the branching is just after 8 to 12 residues. So the branching is highly we can say more and more as compared to the branch points are repeating many times after the interval of 8 to 12 monosaccharide units in case of glycogen. So this is the second thing. So glycogen is stored in liver and is a source of energy. You know it. It is being converted into glucose with the help of hormone known as glucagon. Understand? Then that's all about. And this glycogen will give red color with iodine. It will also give, like amylopectin, it will give red color with iodine and can be digested by both alpha amylase and beta amylase. All right, that's all about glycogen. Then we have one more thing which we call as cellulose. See, this glycogen was a storage polysaccharide because it was storing in our body. But we have the cellulose, and this cellulose is a structural polysaccharide. And you know it is a component of cell wall. It is a component of cell wall of plants. Therefore, we have large number of flora as compared to fauna. So we have more cell walls because plants are abundant everywhere. We have more plants and we have more cellulose in the world. Therefore, it is the largest, or it is the most abundant polysaccharide among all polysaccharides. This is the most abundant polysaccharide in the world. Second thing, when we see this cellulose has a linkage of, it is made up of the monomeric units of glucose. They are repeated time and again. We have a repetition of glucose units. But here the linkage is beta 1,4 glycosidic bonds. We have beta 1,4 glycosidic bond. Beta 1,4 glycosidic bonds is there. There is a single difference. Just the OH should have been below, it is just on the above side. And by this difference, the cellulose is formed. If we just check it here, we have this way, this is the, suppose this is the beta glucose, OH will be on this side, and we have one more, that's beta glucose. Again, the OH will be on the upper side, this way. And this is the continuum. If we see, this is here OH, this is the OH, and here we have the OH. This we have CH2OH and CH2OH here also. You can know it. This is OH here, this is OH here, this is OH here. And how we can make a beta 1,4 glycosidic bond between this OH, this is at position number 1, this is at position number 4. At this, what is happening? This OH and H will go, and here we have O. This type of linkage is known as beta 1,4, and this thing will be continuing. N times this will form what we call as cellulose. So cellulose, by way of the beta linkage, by way of just beta presence of beta 1,4 glycosidic linkage, cellulose, there is no enzyme in the human body or in the human body which can digest the cellulose. So it cannot be digested by alpha amylase. It cannot be digested by beta amylase. One more thing. Can't be digested by the enzymes like amylase. However, you will see cellulose digestion takes place in termites. They feed on the bark, and they are taking the cellulose, they are taking the bark, and they are taking the cellulose. There is some digestion because they contain microorganisms. These insects contain microorganisms. By these microorganisms, they may be digesting the cellulose. And cellulosic digestion is a prominent feature of ruminants, cattle, sheep, we can say cows, buffaloes, sheep, and goats. It's a prominent feature in case of ruminants because they contain microbes in their stomach, the largest compartment of stomach, which we call as rumen, which we call as rumen. Which we call as rumen. And this rumen contains large number of bacteria and protozoa. These bacteria and protozoa are basically this fermenting the carbohydrate, or they are utilizing this cellulose. So microbial, we can say there's a microbial fermentation of cellulose is taking place in case of ruminants. After this, we have the chitin. Again, a homopolysaccharide. Chitin is also a homopolysaccharide. We'll see it here. We have chitin. Some people believe it is a heteropolysaccharide, but no, it is a homopolysaccharide. It is a homopolysaccharide, and it is made up of monomeric units, and the monomeric units are N-acetyl glucosamine. Glucosamine is the monosaccharide units. Therefore, if the repeating unit is of single type, and single type is what? N-acetyl glucosamine. This is the N-acetyl glucosamine is the monomeric unit which is repeating time and again. So you can say this is not a heteropolysaccharide, but is a homopolysaccharide. And here the linkage is beta 1,4 glycosidic linkage. Glycosidic linkage, because it is the N-acetyl glucosamine is exhibiting himself in beta form. If we have to make it, we can just see. We can just show you. This is you make just beta D glucose. You make this is beta D glucose. Here you have the OH, and here you have OH. Here you have OH. Here we have CH2OH, right? And at position number 2, you have an acetyl and amino group. 1, 2, 3, 4, 5, and 6. At position number 2, this OH is replaced by an acetyl and amino group CH3CO and H2. This we can say is the, it is not to be written in this way. We can write it in this NHCOCH3. So here we have an acetyl group and amino group at position number 2. Here we will be having H, and we call it as N-acetyl glucosamine. A repetition of this. Again, we can make one more just to make the illustration. We can make one more N-acetyl glucosamine. it is beta, which is on this side. Here we have NHCO, CH3 group attached to this, N-acetylglucosamine, which on the inner side, on this side, and here we have CH2OH. Alright? So between the two, 1 and 4, this position, the position is same. Here we have 1, 2, 3 and 4, 5 and 6. Between 1 and 4, there is formation of glycosidic bond. This H and OH will go, and this oxygen will be used in the bonding, which we call as glycosidic bond. A glycosidic bond will be formed. This you repeat n times, you are forming what we call as chitin. So chitin is a homopolysaccharide, and we know the chitin is produced by arthropods, kingdom Arthropoda. They are most abundant animals, and because of the presence of hard exoskeleton, which we call as chitinous cuticle. So this chitin is forming the exoskeleton of arthropods, and about 1 billion tons of chitin are produced in the biosphere every year. That's also one more important function about this. We have one more polysaccharide which we call as homopolysaccharide, inulin. Inulin is a fructosan. It is a fructosan, means it is made up of, it's a homopolysaccharide, only one type of monomeric unit, and the monomeric unit is fructose. Inulin is a fructosan made up of monomeric units which we call as fructose. It is made up of monomeric units which we call as fructose, and the type of linkage is beta 2,1 glycosidic linkage. You can make it in the fructose. At one side we have beta, it means position number 2 and then 1, beta 1,4 glycosidic, beta 2,1 glycosidic linkage is shown by the fructose, by the inulin. Inulin again is a homopolysaccharide, and this inulin you will find it present as a reserve carbohydrate. It is seen as a reserve carbohydrate in the roots, in the tubers. You will find it as a reserve carbohydrate in the roots, in the tubers, and in the rhizomes. For example, dahlia. You will find it as a reserve carbohydrate, storage carbohydrate, a reserve carbohydrate type. You will find this inulin is present in the roots and tubers. And this inulin has one very important significance. The inulin, there is no enzyme in our body which digests this inulin. There is no enzyme in our body that's going to digest the inulin. So therefore, and this inulin is excreted out by the kidneys. It can be excreted out by the kidneys. Therefore, this is used for determination of glomerular filtration rate. Therefore, we can understand how much amount of inulin we can give. The whole amount has to be excreted out by the kidneys, filtered out by the kidneys, and excreted out by the kidneys. And we can assess one of the kidney function, that is glomerular filtration rate, by giving inulin and calculating the GFR. That's all about the polysaccharides, and in the next video tutorial we will take it to polysaccharides. Thank you very much for watching.